Fundamentals of genetics. Genetics is the study of how characteristics are actually passed from parents to offspring. So just like you look like your parents, these pictures of these two families, you can see how the daughter looks just like her mother with a few differences, and then you can see all the similarities between the son and his father. And that is due largely to genetics. Heredity is how traits are passed from parents to offspring. So just like you probably look like your parents, it's because of how things are passed on. And we just talked about cell division, about how those genetics and how the DNA is passed from cell to cell. This is exactly why you look so similar to your family members. So the person who really is credited with figuring out a lot about genetics is Gregor Mendel, and he was lived in the 1800s, and he was at an Austrian monastery, and he was the gardener for the monastery. So he started to work with pea plants, and he noticed different ways that the plants passed on traits, like the color of the flower and the height of the plant and the type of the pea that was produced, if they were either wrinkled or they were smooth. And so he started running tests, and he bred different plants together to see what kind of traits resulted. So he's actually credited with being the father of genetics, and he's really important to the study of genetics. A lot of what we do today is because of him, especially with figuring out how traits are passed on to offspring. So he observed 7 main characteristics of pea plants. He only looked at traits that had contrasting appearances. So he looked at, for example, flower color. The flowers were only white or purple. There was no in between. Same thing with the height. So they're either tall or short, yellow or green. So he looked at contrasting traits. And he used statistics to study these over many generations. And the good thing about pea plants is that they quickly breed, and every season you'll have a new crop of plants. So he didn't have to wait very long to get results. So here's some examples of what he found. So these are the traits that he looked at. And so he controlled how they were pollinated, so he knew exactly who the parents were, and then from those seeds, when he planted those, he knew exactly who the parents were, and so he could predict what those traits might be in the offspring. He grew plant strains that were pure for a certain trait, that always produced a certain trait. So for example, he produced plants that maybe always produced a purple flower no matter what. They always produced a purple flower. Same thing with the other traits. So he crossbred first 2 pure strains of contrasting traits. So for example, he crossed plants that always produce purple flowers with plants that always produce white flowers, and he called that the F1 generation. And then he allowed that F1 generation to self-pollinate. And then he called the resulting generation the F2 generation, and these are important. So this is what he did. So he has at the top, you can see the purple, which is big R, big R. So there are 2 dominant traits, and always produced purple flowers. And then the white flower is little r, and so they always produced white flowers. We're going to talk a lot more about those letters in a little bit. And then, so when he did that, he noticed that all of the F1 generation turned out to be purple. But during the self-pollination process, where the F1 generation bred, then he noticed that the white trait came back out, and he really thought that was cool. So he wanted to go on with that and see why in the F2 generation the recessive trait became visible again. And this is what he figured out. So the first thing he said was that they were dominant and recessive traits. A dominant factor, so remember we talked about chromosomes, and we said that there are 2 homologous chromosomes, right, per pair, and those homologous chromosomes have the same genes located on them, and this is what we're talking about. So there are 2 alleles. There's an allele on one homologous chromosome, and there's another allele that codes for the same trait on the other homologous chromosome, and those alleles can be dominant or recessive. Well, the dominant one, just like you might think, the dominant will mask the recessive, so it kind of takes over, and the recessive is kind of the one that is like the white color. So if there's a dominant allele, you're not going to see the white color. He also said the law of segregation. So he said that the pair of factors of the alleles are segregated or separated during the formation of gametes. So each gamete only receives one factor. So just like when we talked about sperm and eggs, the sperm only contains maybe just one of those homologous chromosomes, so one of those alleles. And then in the same way, the egg contains another allele. So when they come together, they'll have two alleles and they'll have different traits on those alleles. So one might be dominant, the other might be recessive. You might have two dominants, you might have two recessives, just depending on what the sperm and the egg received. And he also talked about the law of independent assortment. So these are both Mendel's laws. And he said that factors of different characteristics are distributed to the gametes independently. So remember, gametes are sperm and egg. So he said that whatever is passed down isn't really dependent on um, anything else but just pure chance. So you might, if you have an allele, a dominant and a recessive allele, when you produce sperm or eggs, those might have dominant or recessive. There's not really any pattern to that. It's just a random assortment of alleles. In molecular genetics, that's when we talk about the structure and the function of the chromosomes and genes. We already talked about chromosomes. They're like the thread-like structures made up of DNA. And the DNA is actually wrapped around proteins and coils up really tightly into those rod shapes. And then we have already talked about genes a little bit too, but remember, genes are segments of DNA in a chromosome that actually controls a trait. So, for example, you might have a gene for skin color. You have actually several genes for skin color. A gene that controls eye color, a gene that controls if you have a widow's peak or not, or attached earlobes. And then alleles are those alternative forms of a gene. So like we talked about before, you have a gene that codes for eye color. Well, that might be a dominant allele or a recessive allele, and you have two of them for each trait. These definitions are very important, and we'll talk about these a lot. I'll use these terms a lot and then in the upcoming lectures. So a genotype is just the genetic makeup of an organism that only talks about the DNA, the different alleles that an organism has. Just like we talked about with the flower colors, we had big R, little r, big R, little r. The phenotype is the physical appearance, and that's how you can remember it. Phenotype, physical. And that's what you can see. So you cannot see a genotype, but you can see a phenotype. So a phenotype is largely dependent upon the genotype, but environment also plays a role. So a phenotype would be like purple flowers, brown hair, wrinkled, spotted, that kind of thing. And then we have homozygous and heterozygous. So homozygous is when both are alike. Remember we talked about homo as a root in biology means always means the same. And so homozygous, homo would mean all alleles are alike. So it would be little p, big P, big P. And then heterozygous, that they're different. So you have a big letter and a little letter. And then probability is just the likelihood that a specific event will occur. And we'll talk about a lot about probability when we look at different traits and how they are passed down to offspring. You'll see the probability changes depending on the types of dominant and recessive alleles.